Good morning and good afternoon, as the case may be, to everyone who's joined us. Welcome to today's X-Talks webinar. Today's talk is entitled A Novel Approach to Applying Genomic Patient Data to Improve Multiple Sclerosis Treatments. My name is Megan Laurance, and I'll be your host for today. I am the Senior Director of Data Services at DNAnexus, where it's my role to work with healthcare institution partners and pharma clients to build disease-specific Clinico-Genomic datasets that are designed to accelerate precision health and biotech and pharma research and discovery goals. Today's webinar will run for approximately 60 minutes with time for question and answer session at the end. This webinar is designed to be interactive, and these webinars really work best when you're involved. So please feel free to submit questions and comments for our speakers throughout the presentation using the questions chat box. And we'll try to get to your questions during the Q&A session. This chat box is located in the control panel on the right-hand side of your screen. If you need any assistance, please send a message using the chat panel at any time. At this time, all participants are in listen-only mode. Please note that this event will also be recorded and made available for streaming on xtalks.com. At this point, I'd like to thank Sutter Health, who developed the content for this presentation. From physician offices to hospitals to outpatient care centers and home services, Sutter Health proudly supports more than 3 million people, nearly 1% of the US population, in one of the most diverse and innovative regions in the world. Sutter Health is more than 60,000 people strong, thanks to its integrated network of physicians, employees, and volunteers. Rooted in Sutter Health's not-for-profit mission, these team members partner to deliver exceptional care that feels personal. Sutter team members adopt new technologies, make novel discoveries, and embrace creative thinking to help patients and communities achieve their best health. Our speaker for today is Dr. Greg Tranah. Greg is the Executive Director of Precision Medicine at Sutter Health and an adjunct professor in the Department of Epidemiology and Biostatistics at the University of California, San Francisco. At Sutter Health, Greg directs the Precision Medicine Center dedicated to accelerating the discovery, translation, and clinical implementation of genomic predictors of disease progression and treatment response for patients across this 3.5 million patient healthcare system. In addition to building a 100,000 patient biobank representing several disease areas in oncology, cardiovascular disease, aging, neurology, and movement disorders, Greg is developing a research program integrating electronic health records and genomic data to transform clinical practice and improve patient health and functioning. And now, without further ado, I'm going to hand the mic over to you, Greg, and you can begin whenever you're ready.
Oh, thanks, Megan. I've been looking forward to this. So everybody hopefully can see that we are going to both be presenting today's talk. So you'll hear both Megan and me talking. And this is an exciting opportunity to share the work that we've been doing together, because I think this is really a way forward for Sutter Health when it comes to doing precision medicine research, and also for other healthcare systems who are developing similar programs or at least thinking about it. So here's the agenda for today. We'll briefly talk about challenges when it comes to multiple sclerosis specifically, and precision medicine research more generally when it comes to doing this type of research in a healthcare system, how we're partnering to overcome these challenges, and specifically as an example, the Clinico-Genomic study we're doing in multiple sclerosis, because there's a lot of work to be done. And then finally, where we go next with this. So after we've launched this project and are starting to move it forward, how do we take this example and move into other disease areas that are critical importance to our patients and to the world? So for multiple sclerosis, the prevalence of disease is growing. It's more than doubled since the 70s, which is the last time there was a serious assessment of MS, at least in the US. At the current time, there are about 1 million MS patients in the United States alone and over 2 million worldwide. You can see that the global distribution is largely latitudinal. You can see higher risks at the higher latitudes, and those risks diminish as you get closer to the equator. MS is a disease that is much more common in women than it is in men, about 2.8 times higher. And MS risk also varies dramatically by race, ethnicity, socioeconomic status. And so these are all challenges that when it comes to doing this type of research, we need to understand these factors as well. There are three main types of multiple sclerosis that are recognized. The most common is known as relapsing remitting multiple sclerosis, and this is characterized by short durations of relapse and which are interrupted by symptom-free times for any time from months to years. And so you can see the pattern on the left where a patient may be experiencing disability and then have that disability remit, and then later on there's relapse and so on. There's another form that is not as common as relapsing remitting. It's called secondary progressive, has similar patterns of relapse and remit, but the relapses don't fully remit. So meaning there's more there's more degradation, there's more disability over time. And then finally, the least common but most devastating is primary progressive, where there is no relapse time. It's basically a rapid decline to death, and it's really devastating. And this accounts for about 10 to 15% of overall MS. The treatment landscape is interesting and is rapidly evolving. And treatments are really designed to either modify disease or disease-modifying therapies. There are also other ways of managing relapses, as well as managing symptoms, and this does not have to involve disease-modifying therapy. There are other ways to manage relapses and symptoms. When it comes to the approximately 17 DMTs, there's a wide range of administration, oral, injection, infusion, and there's a wide range of effectiveness and toxicity when it comes to these drugs. And this is why it's really important to understand what a patient's disease course is going to be over the lifetime of their disease, as well as how severe, and as well as understanding what their individual response to a drug would be, so we can understand when it's better to avoid a more toxic drug. And so that's something that we'll come back to later in the talk, but this is why it's so critical to do this research. The current state of genetics in MS is nicely summarized by this paper, but there is a nice history of genetic research in MS. To date, there are approximately 200 genetic risk variants that have been recognized from genome-wide association studies. These account to maybe up to half of the overall heritability of MS. And these loci are all involved in acquired and innate immune cell function. And so this is a very nice place for us to be starting our genetics work, because there's such a solid foundation from large cohort and population-based studies. Yet there remains this discovery gap, which I imagine a lot of people on this presentation might already understand. So we're targeting identifying rare variants in the genome that are related to disease onset, disease progression, as well as treatment response. Determine if there are other pathways involved in disease outside of immune cell function, and then establish or at least identify associations with onset and progression, whether or not we can actually tell the different disease subtypes apart earlier at the diagnosis stage, and determine whether or not there is a predictor of treatment response, and if we can understand clinical phenotypes better, whether it's MRI or other movement and clinical phenotypes better at the genetic level. When it comes to MS as a disease and the etiology of it, there's, again, a pretty healthy literature on differences by race and ethnicity, age and sex, latitude. There are also modifiable risk factors like socioeconomic status and behaviors, whether it's diet and exercise, that are already published. But again, there remain gaps that we're addressing at the clinical level and in our patient population to identify whether there are other phenotypic drivers of disease, what the genetic drivers are, and if there are genotype-phenotype interactions that might be causing some of the burden of this disease. And again, we're bringing deeper phenotyping to this project, so we have a wealth of MRI data and other clinical phenotyping that will be used in the data set that we're building together.
So thanks, Greg. So I think what Greg really nicely summarized are the specific, with giving a great background on kind of the state of the field in MS research, but with a specific eye toward the challenges in precision medicine for multiple sclerosis. One of the things that he highlighted is just a gap in understanding in certain areas, whether it's a need for deeper phenotyping or need for deeper genomic data. And those challenges were certainly challenges that we at DNAnexus were hearing across our extensive network of academic medical center customers, pharma customers, who, as they were adopting our platform for analysis of data, it was very clear that everyone is struggling with access to the type of data that will support these types of precision medicine queries and research goals. And so not just in MS, but across the precision medicine endeavor, so to speak, there's a real need for access to broader real-world clinical data to gain deeper insight into the heterogeneity of disease, the difference in patient outcome, the difference in onset, et cetera. A real need for deeper genomic and phenotypic data to understand how each of those axes of the data contribute to disease onset and progression, and also drug response. And all of our partners and customers who are involved in these precision medicine or learning healthcare initiatives are looking towards this data to help transform the treatment of disease and ultimately improve patient outcomes. We also were hearing very strongly among our pharma customers that they were embarking on a longer-term strategy for utilizing real-world data in pursuit of regulatory-grade real-world evidence. And so given all of these challenges, again, not just in multiple sclerosis, but across our network of partners, we realized that there was a real need for disease-specific data sets, because those that were out there to fuel this research either didn't exist at all, or they fell short in genomic or clinical detail, or were incomplete, or the quality wasn't necessarily as robust as it needed to be, or in some cases there were great data sets on both genomic and clinical scale and axes and detail, but they were inaccessible to commercial entities. And so in a sec, we're going to talk a bit about how forming this partnership between Sutter and DNAnexus is helping to address that data gap. First, I want to take just a minute or two to explain a little bit of background on real-world data, so-called real-world data, and just kind of level set on what generally the accepted definition of real-world data is, because that really is one of the unique components of this study that we're launching, and of all of the studies we'll be launching with Sutter and other partners. So the reason that we're going after real-world data is that it will enable us to get a much more extensive and representative collection of data around a real-world MS population. Generally speaking, real-world data refers to any data that's derived from the patient experience outside of conventional clinical trials. And so the strategies that those that are looking to access real-world data or generate real-world data are adopting include harvesting both structured and unstructured data from electronic health records that will enable us to establish a natural history of the disease. Also, this will enable us to create a repository of longitudinal patient data, including treatment courses, disability progression, and understand therapy response in patients with comorbidities who are almost universally excluded from the clinical trials that are used to establish drug response. And so, in particular, our pharma customers are very eager to understand, how these disease-modifying therapies in MS are performing in the real world, not just in tightly constrained clinical trials that exclude a more broad and representative MS population. Also, real-world data utilizes data that is not going to be present in the electronic health record because it's outside of standard of care. That includes patient questionnaires that gets at extensive family history, and we'll go into the details of that in a minute, but also the opportunity to collect patient-reported outcomes, all of which helps fill gaps in data that's only recorded during routine clinical care. And so overall, this strategy of collecting and integrating real-world data into a Clinico-Genomic data set will help us represent the patient experience in a much more real-world patient experience and population. And so for the next few slides, we're going to talk about how, when looking at all of these challenges, the gaps in data, the need to analyze this data using a robust platform, as we were looking to help solve these problems for our partners and our customers, we already had a relationship developing with Sutter, and we had a clear opportunity to help address these challenges through a partnership. And so we're going to talk for the next few slides a little bit about Sutter and a little bit about DNAnexus.
Great, thanks, Megan. So here's a slide of Sutter's place in the world. You can see the map shows our region of Northern California where we treat patients. We are the 4th largest integrated delivery system in the US with more than 3 million patients per year. It's about 1% of the US population. And hopefully you can recognize from this map that this is one of the most diverse places in the world. Demographically, and as well as when it comes to patient treatment options, when it comes to the diversity of diseases we see, the staging of disease. So, we see it all with our patients, whether it's early stage or advanced disease, whether or not patients are treatment naive or whether or not they're on their 4th or 5th round of treatment, we see it all. And this is what I like to think of as the real world. We have over 5,000 physicians, 28 hospitals, and ambulatory surgery centers. And one of the most interesting things I think Sutter has done is invested in producing one instance of Epic, which goes back a decade or more in some cases. And this allows us to do research at the system level, so we don't have pockets of data that are highly localized. We can leverage the entire system for doing research. Within Sutter, we've invested in developing a large research enterprise, which means scaling our clinical trials to the system, the ability to do more informatics and digital health research and bring those solutions to patients, as well as a large precision medicine initiative. And my job with this initiative is to identify what stages they are most likely to experience, and that will help us understand what interventions will work best for a particular patient at a particular time. And so 2 large initiatives I like to use as a way of demonstrating how we're already involved in leading this type of research is our large biobank. We are biobanking 100,000 patients from across our system, collecting blood, serum, and plasma longitudinally. We're focusing on our service line strategy for targeting which patients we want to help using this infrastructure. This includes neurology, oncology, cardiovascular disease, as well as human aging. And with these longitudinal samples linked to the electronic health record, we'll have a tremendous asset for leading research related to biomarker discovery, so we can, again, help our patients and patients beyond our healthcare system. We also have a very strong basic biology infrastructure where we're working with patients that have the most treatment-resistant and devastating cancers. We're able to remove tumor tissue, bring it to the lab, and do a number of laboratory screens on the genomic and drug screen level to then bring that back to the patient and their physician to optimize treatment. So this is something that we've been involved in for quite a while. And when it comes to oncology, we have more cancer cases than a lot of the academic research centers in this area combined. So this really means that in order to accomplish this research at a large scale, we need a solution for bringing data together from the patient level, from the genomic level, so that our physicians and our researchers can make the best use of it.
Thanks, Greg. So as we started just early conversations around forming a partnership to start sourcing all of this data and collecting it and integrating it, it was just really clear from Sutter's really diverse and extensive population, plus the opportunity of having a single instance of an electronic health record, such as EPIC, all sorts of pieces were falling into place. And so, as Greg mentioned, what DNAnexus is really bringing to this partnership is a platform that will host, ingest, harmonize, and enable the analysis of all of this data. And so just at a very high level, DNAnexus has, for the last 10 years, we're about to celebrate our 10th year, has really been focused on solving our customers' challenges around analysis of very large-scale genomic data, and more recently, very large-scale combined multidimensional, multi-genomic linked clinical genomic data sets. And so really the best way to think about DNAnexus as a company is our focus on what we provide to the life sciences research field is threefold. The platform, which I'll talk about in a little bit of detail in a sec, our extensive in-house scientific expertise, really putting the science first and foremost, and our customers' scientific challenges at the forefront of everything we do, including services and the products that we build and launch. And also the fact that this platform has been out there for 10 years has enabled us to build essentially a global research network with the opportunity to connect all of this data. But really at the core of what we do is the DNAnexus platform, which is a scalable, secure, and compliant cloud-based platform for analysis of complex data sets, really with an eye towards really high velocity computing. Many of our customers have hundreds of thousands of samples to process, trillions of data points when you combine the genomic and the clinical data. And also the layer of security and compliance that is embedded and integral to that platform that really gains the trust of our partners, such as Sutter Health, in trusting us with the data that comes to the platform. So I'm going to spend just a couple of minutes talking specifically about one component of our platform, and that is a more recently launched solution called DNAnexus Apollo. DNAnexus Apollo was very intentionally built to address these complex multidimensional data sets where our customers, whether that's an academic medical center or a healthcare provider such as Sutter or a pharma company, are dealing with multiple clinical data sets that have linked genomic data set as well, and also have teams of scientists, both biologists and bioinformaticians and computational biologists, that all want to be able to explore and analyze this data collaboratively and critically in a very secure and scalable environment. And so we built DNAnexus Apollo specifically to address that use case of large-scale, multidimensional data sets where there's really teams of experts that are attacking the data from multiple angles. And so specifically what DNAnexus Apollo supports, if you look at the top left, what DNAnexus is well established in this space for analysis of large-scale sequencing data. And so DNAnexus Apollo is designed to ingest all of that large-scale sequencing data, whether that's whole exome sequencing, whole genome sequencing, or multi-omics through RNA-seq, et cetera. Then link that genomic data with clinical data. In our case, also for this MS study, linking it with imaging data. And for many of our customers who already have access to publicly available data sets in the same field, there's a strong need to integrate all of this data onto a single platform that is easy to use for biologists and has the deep programming capabilities that are necessary for the bioinformatics computational biology component of these teams to enable global collaboration, and the very familiar to all of you researchers who are listening in today, a platform that will enable this ongoing cycle of discovery and validation, hypothesis generation, testing, sharing, and then validating those insights. And so the way that DNAnexus Apollo supports this life cycle of research is by building a number of different features, releasing a number of different features that after the data is ingested, enables carving out specific cohorts, browsing the Clinico-Genomic data, various dashboards and visualization, algorithms and statistical analysis. Really importantly for our technical users, the ability to launch their own algorithms and programs and applications using an embedded Jupyter notebook in the cloud, and thereby supporting, again, this multi-dimensional, expertise team in the analysis of these really complex data sets. And so really quickly, just a glance at what the cohort management and dashboard and visualizations look like. Again, the ability, once the data is ingested and harmonized into the DNAnexus Apollo data model, biologists and bioinformaticians alike have the ability to browse through these cohorts and carve out subsets of the data set that meet very specific genomic and phenotypic criteria. And from there, launch analyses either through out-of-the-box statistical analyses such as GWAS or PheWAS that are allele frequency calculators that are, again, available out of the box, or through the technological team members, bioinformaticians who have designed and launched specific algorithms and applications within the DNAnexus Apollo infrastructure, again, for extensive statistical analysis and getting at all those correlations that Greg was speaking about earlier. Are there phenotypic components that correlate with progression or early onset? Are there genotype correlations between treatment response, et cetera? And so all of this partnership, what Sutter contributes and what DNAnexus contributes, adds up to an overall solution that we think will be extremely valuable, not only for MS precision medicine, but for other disease areas as well. And I'll let Greg talk a little bit more about that.
All right, thanks, Megan. And so everything you described allows us at Sutter, our physicians, our researchers, to basically accelerate discoveries related to both our patient data as well as the genomic data that we're generating. So this allows us to have cutting-edge tools, which we don't currently have, and that's why we have this partnership. And so what we bring is access to, again, a massive, diverse patient population, the diversity in disease staging, treatment history, and outcomes. And I think one of the most important pieces to all of this is we have an existing strong patient right and data protection governance protocol. And this is reflected in our extensive consenting process, RD reviews that we undertake for any studies like this. and our data protections already exist. So, we're just bringing that to this project. Also, being in such a large healthcare system, we have a continuous source of very rich, complete data longitudinally, as well as retrospective data, and the ability to interact with patients as disease progresses or not, and as outcomes are experienced. We also partner closely with our physicians and our patients, so we can administer questionnaires, add additional clinical measures such as the EDSS for multiple sclerosis, and these allow us to fill gaps in the electronic health record because there clearly are gaps, and a lot of the data we need aren't easy to obtain. So we by partnering with physicians and patients can fill those gaps as needed. And with our expertise in data capture, we are able to extract data from our EPIC EHR, as well as undergo chart review and combine all these data for retrospective and prospective follow-up. And then finally, we have a system-wide infrastructure for collecting and processing biospecimens. So this allows us to really biobank at scale. And it's all of these assets that make us very excited about doing the research I've described. It's just we don't have the informatics solution, and that's why this partnership makes so much sense for us. And that's why on this next slide we call it the solution, because this is now the specific study, the specifics of the Clinico-Genomic data that we're producing. And this is what it looks like. Looks pretty straightforward. We're combining genotype, phenotype, imaging data to store in the DNAnexus cloud securely, safely, in ways that are compliant with our patients and institutions' needs. All of the blood that we collect from our participants goes out to the University of Pittsburgh Medical Center for whole exome sequencing in the DNAnexus platform that is combined with retrospective capture and longitudinal follow-up from a number of sources within Sutter. And then finally, we're adding additional data when it comes to imaging, EDSS, and questionnaires, which we'll get into the details a little bit. So now the map is back, but this is to illustrate where we're now going into the clinics to do this study. So at Sutter, we have over 3,000 MS patients across Sutter Health. Our goal is to enroll more than 500 patients, hopefully up to 1,000. And we're starting with our 5 largest neurology clinics where a lot of our MS patients are seen. These are indicated on the map. So we're consenting patients in Sacramento, in Berkeley, which is our first site that's opening up. We're also down in the South Bay in Palo Alto, Mountain View, in Santa Cruz. And this again allows us to capture the diversity of MS when it comes to disease staging and treatment history, but also the diversity of patients, whether it's race and ethnicity or socioeconomic status. This is where our patients come for care, and this is how we're going to do leading research in this area and to understand the diversity of disease. And even though our primary focus is on patient care, we do have experienced clinicians and clinical coordinators across the system that make this research possible. And so we've been involved in a lot of large research studies already. We conduct lots of clinical trials, so this is not new to us. This large-scale genomics research is what's new to us. And we have existing data analytics support at Sutter Health. So again, expertise in getting into the electronic health record and going through charts to get to the data that we need. So this is what our initial study visit workflow looks like. By having EPIC, we're able to identify eligible patients as they come into the clinic or are making appointments to come into the clinic. This way we can staff a clinic and even reach out to a patient ahead of time. And that way we can maximize the days where there are the most patients in a clinic and try and really help as many people as possible. So at the initial study visit, we will consent the patients. We will have blood drawn. There is a questionnaire for the patient, and also we'll perform an EDSS, which is a disability assessment. All of those things, when combined at the DNAnexus level, will allow us to do the types of research we've been talking about all along.
Thanks, Greg. I do want to say that while this is not an interventional clinical study, it is an observational clinical study, and so Sutter has been a tremendous partner in ensuring that we are keeping the patient at the center of this process. And so as Greg just walked through that initial study visit workflow, again, we are asking patients for a blood sample, we're asking for their time to fill out a questionnaire. And one of the huge benefits of working with Sutter is, again, their real focus on truly informed patient consent and keeping the patient at the center of this process. And so I'm going to pick it up from the point where once the patients are consented, we will have permission to do a very deep dive into electronic health record data, doing a full retrospective capture with longitudinal follow-up. And again, the advantage of there being a single instance of EPIC at Sutter Health for 10 years means that so long as the patient has been getting their care at Sutter Health, that retrospective data will be extremely rich from the very beginning and from the get-go. So after that initial study visit, we will start the process of extracting clinical data, patient data from the electronic health record, and we'll be using multiple methods for that. One is, again, connecting with the data analyst experts at Sutter who know that EPIC data really well, know which data elements are complete, which are notorious for false positives, which need to be interpreted in a particular way, and start extracting structured data elements from the electronic health record database. It will also, once patients are consented, will kick off the manual curation process, which is necessary for a number of really high value data elements that either are not available in the structured data, so they're buried in progress notes or imaging notes, or just aren't are embedded in a PDF from a biomarker test that may have been done that's also really critical. And so really what this slide is meant to depict is that this data extraction and data collection starts with retrospective capture, harvesting as much as we can that we know is critical to supporting these precision medicine and learning healthcare use cases. But we will also be longitudinally following these patients for a minimum of 4 years. And this is really, this process is what's going to enable us to establish this natural history of the disease with a strong focus on disease progression, disability measurement, and response to treatment. So all of this data will be collected in Sutter's electronic data capture system, again, either in an automated way or through manual curation. And I'm going to go into a little bit more detail on all of these clinical data elements, just so you get a feel for the categories of data that we're going to be collecting. One thing I will add that I think, in addition to the platform being something that DNAnexus is really bringing to this partnership, is just the fact that we have been building and launching and releasing products with a very intentional process of making sure that what we are building meets customer need and addresses the use cases. And so with such a rich wealth of data at our fingertips here, it's really easy to go overboard and extract either way too much or not extract enough. And so what DNAnexus, in addition to the platform, brings is the ability to operationalize this process, so to speak, so that we are really understanding which data elements are critical to support both Sutter and third parties in their research on this data. So with that in mind, we're certainly focusing on collecting demographic information, fairly straightforward for this type of data set. A very deep dive on medical history. Again, whatever we can get through structured data elements, we will, and we will be supplementing that with data collected through patient questionnaires. Everything covering other autoimmune diseases, not only in the patient, but in extended family, health behavior, healthcare utilization, et cetera. Also collecting a lot of laboratory data that's used, again, to establish this natural history of disease, either disability progression or just basic health measurements, or vitamin D, known biomarkers, and particular lab values that are very high interest to MS researchers. We'll also be collecting imaging data in the form of DICOM files from MRI images, and through the manual curation process, pulling qualitative imaging data from interpretation notes. Moving on, so other areas that we're collecting data includes capturing that really critical understanding of which disease subtype the patient has. Again, this might not be a standard of care note in the electronic health record, so we're covering all our bases and adding that to a physician questionnaire as well. One area that is really of critical value and interest is a full understanding of prescription and medication history. This is an area where we can get quite a bit of data from the electronic health record, but we also know we need to fill in some of the holes on that complete treatment regimen through patient questionnaires and scouring through progress notes as well. It's really critical to understand reason for discontinuing a particular disease-modifying therapy when one was ended and one was started. We'll also be capturing a number of outcome measures around disease and disability progression, some of which will be captured from unstructured data, such as progress notes. The neurologist conducted EDSS will actually happen at the initial study visit. If there was a 25-foot walk or other disability assessment that was done during routine care, we'll capture that from the notes as well. And also some extended questionnaires to get a quality of life, fatigue, cognition assessments, et cetera. So as you can see, it's really quite an extensive data set that we're going after, but again, with some discipline around making sure that everything that we're capturing will support a specific use case. And so DNAnexus will be contributing to this process. So all of this stuff will be happening on site at Sutter. Their expert chart abstractors, their expert data analysts will be pulling the data, curating the data. All of that will be happening on site through Sutter's, again, data teams and study nurses who will be collecting the data, and will all be collected inside Sutter's electronic data capture system. And that data will go through a number of quality checks, adjudication, et cetera, will be deposited in a study-specific database at Sutter, and then will be exported and adjusted into the DNAnexus Apollo platform, where once it's matched with the sequencing data, will enable all of that discovery and analysis. And so, as Greg mentioned, at that initial study visit, a blood sample will be collected. One of those samples will go to UPMC's Genome Center, where samples will be processed, libraries prepped, and whole exome sequencing performed on NovaSeq, which gives us tremendous scale and speed and accuracy and depth of sequencing. Those sequencing files will then, that raw data will come to DNAnexus, where our expert team of in-house bioinformatics expertise and scientists will run analysis pipelines to move that data from FASTQ to BAM to called variants using DNAnexus really robust analysis pipelines. And then it all comes together on the DNAnexus platform, the genomic data, the clinical data, the imaging data, and that's where the discovery using Apollo will begin.
Thanks, Megan. And so for us, what this means at Sutter Health is this is the first time such a wealthy, deep data set will exist for our patients in our system. And this will allow our clinicians and our research teams to access these data on a platform that is friendly for people who may not have experience with genetic analysis, and allow us to visualize, analyze, generate knowledge from the data, whether it's we're trying to understand genetics of subpopulations, of disease types, if we can model treatment pathways or even understand outcomes. And so this platform enables us to be big players in this research, because at the moment, while we have access to the biospecimens and the patients and can securely analyze those data, we don't have the ability to integrate with genomic data and analyze.
And so as this all comes together, one of the things that was really critical in setting up this study was ensuring that our informed consent, and as Sutter conducts the initial study visit, that through that informed consent, patients understand that their de-identified data will be accessible to third parties. And so the section on the right really describes, so DNAnexus is making an investment here, Sutter Health clearly making an investment here, but pharma and biotech companies will have the opportunity to license access to this data through the DNAnexus platform in support of their own research and development programs, where they're really looking towards this combined clinico-genomic real-world data to get a better understanding of disease heterogeneity and outcomes, understand whether there are yet undiscovered genomic drivers of response, phenotypic drivers of response, and also potentially new target identification. And so all of this will happen because the partnership with Sutter and DNAnexus is really strong and in depth, and data governance and providing access to that data through shared parties will also be something that is done in partnership with Sutter, again with patient protection, and really qualifying those third parties who have access to the data as part of the process as well.
And just to make clear from the Sutter side, the investment that comes into this partnership comes right back to our research. So we're recovering our costs and continuing to invest in this research through these types of partnerships. And so what does this at a large, from a large scale perspective, allow us to do as a healthcare system? We envision that this is going to help us become a learning healthcare system. So I think it's pretty obvious the advantages of executing the discovery of information, the validation of that information, and implementing research of that information in a single system has advantages. We all know that discoveries made in a small academic or maybe industry setting don't translate to the real world. And so by transitioning to making the discoveries, validating the discoveries, and implementing changes in healthcare in such a large system makes a lot of sense. And I really want to bring this back to the patient, because this is why we're doing this. And I just want to emphasize what our physicians hope to get out of this and what this means to our patients. So our physicians know that there are no biomarkers, there are no blood tests to understand what a patient's prognosis is going to be or how they're going to respond to medication. And so by understanding the disease at that level, we will do a much better job of identifying safer, more effective treatments for individual patients. And I talk to the physicians in our system, I talk to patients, and this is what they hope for, and this is what I hope to deliver. And the patients just know that the existing drugs are minimally effective, and as disease progresses, quote, little by little, you lose yourself to the illness. And this is why we're doing this. We want to help all of our patients, and by working across our system and partnering, the discoveries we make will hopefully extend far beyond Sutter Health. And so as we wrap up, there are a few key takeaways from the Sutter Health perspective. We've listed all of the amazing assets we have, the ability to assemble large cohorts of patients for disease-specific research, but it's a lot of work. It's not easy because we want to make sure we do it correctly, safely, protect our patients, and only do the research that's going to get back to them and improve their lives. And so designing a research program or a research project, even just like this, requires intense attention to detail when it comes to data governance, making sure that our technology partners are just as safe and secure. So we've already gone through a long privacy and security review with DNAnexus. We need to understand that the data teams, both at Sutter and DNAnexus, understand what the data coming in and what the data going out look like, what the quality and completeness of those data are. And none of this would be possible if it wasn't for the innovative leadership at both, in both partners, allowing us to move forward and develop a program like this. And strong communications within our companies and across our companies and being transparent is critical, because my partners internally are not just the research groups, but also legal, privacy, goes far beyond just the research. So the next steps are to scale enrollment across Sutter to increase the number of MS patients we bring in. We do want to scale the data extraction process and also operationalize to basically expand to other disease areas. So MS is our foot in the door, and once we've made this project happen and get it underway, we're looking for the next projects to launch, whether it's across our service lines in oncology or cardiovascular disease, or in other areas where we have incredible clinical strengths. So with that, I really want to acknowledge all my partners at Sutter Health, and it all starts with the patients who are so trusting and allow us to do this research, whether it's with their time or their samples, or allowing us to make use of their data to make discoveries that will transform healthcare. My partners include the clinic staff at every site that we're going to be consenting patients. And of course, my research teams, I couldn't do it without the A team I have, both on the data side and the clinic side. And my IRB, OGC, privacy and operations partners make sure we do everything correctly and that the patient is protected. And finally, as a not-for-profit healthcare system, a lot of the research I do is supported by our donors. And that's a really powerful part of the Sutter team, is the way we work with philanthropy and the way our donors support the research. So I appreciate everybody at Sutter Health who's making this possible.
As do we at DNAnexus. This certainly wouldn't be possible without the strong partnership with Sutter and just the fantastic team of folks who've been working with on the ground, both in the clinic and in the data centers, all of which Greg just highlighted. I also want to thank all of the work that our data services team and our science and product teams, both on the DNAnexus Apollo and bioinformatics side, who've contributed to our ability to engage in such an ambitious project. So I think that brings us to the end. And Greg, I want to say thanks very much for such a great presentation and taking the time to spend more time on explaining everything that's going on at Sutter and the focus on precision medicine. So now I think many of you have been submitting questions as we've been going along, but I want to invite anyone else in the audience within this last 5 minutes or so that we've got to continue sending your questions or comments using the questions window. And while you're doing that, we've already gotten some questions. And so Greg, I'll start with one of those, which is, how do you plan to scale the study beyond the first phase? You mentioned, going for 500, even getting to that 500 and then scaling to 1,000. How does the scaling of the study work?
Right, thanks, Megan. So this is a really important question, because we all recognize that we're not going to make the discoveries that cure MS with 500 or 1,000 patients. We need to really partner across healthcare systems in order to bring the genomics to a level that allows us to understand subclinical phenotypes as well as disease progression. Within Sutter, we've targeted the first 5 neurology clinics based on the number of patients that they see. The power of our system and our electronic health record is we know where the patients are going to get their care. And so as we roll out to more clinic sites in the system, we will be able to go to the next 10 clinics, which see more patients, and the next 10 clinics after that, which see a lot of patients. And that allows us to reach out to specific clinicians who are seeing patients to bring them in as partners to this research in order to scale internally from the first 500 to 1,000 and beyond. And this is happening in being dovetailed with our biobanking endeavor. So patients who come in are also invited to be part of our biobank. And our goal with the biobank is to get every MS patient who wants to participate into our biobank so they can be part of this research. And just as an aside, as we are outrolling this project, we have patients who are coming to us who want to participate. So it's not just us pushing, we are receiving a lot of attention from patients already.
Thanks, Greg. One other question that came in was around, we mentioned that we would be collecting patient-reported outcomes. The question was, will those patient-reported outcomes be pushed back into the electronic health record, back into Epic, and become part of their record?
Oh, that's a good question. You know, there are cases where we have what we call NeuroShare and other projects where patients are answering questions, whether it's quality of life or how their sickness is going, or if they're responding to treatment or having bad outcomes. And those that information does get right back to the physician. So this information will follow the patient and be available for their caring physician in order to understand better what the patient is experiencing. And in fact, all of our clinicians and our researchers will have access to the data that are being put onto DNAnexus, and those data will be available for their clinical care.
Great. I think we have time for one more question really quick, and actually you and I could both take this one. The question is, can you expand on the scope of the collaboration between DNAnexus and Sutter Health? Is this only for MS?
We're envisioning going beyond MS into other disease areas. We've actually been talking about oncology and other areas. So I'm excited about this project because, again, this gives us access to a platform that anyone in our system can use to do these analyses and learn about genomics and health. And so once this is up and running, and why not, you know, move into other disease areas? And the fact that there's a lot of trust that has already been built, and that our privacy and compliance groups have fully vetted the system and know that patients' data is protected, really is an amazing asset for us.
Yeah, and I'll just echo that by saying that, you know, so much of the hard work of getting this first study up and running just makes the pathways are being established, neurons are firing, and everything is going to be going really smoothly. That enables us to look forward to the next disease area with Sutter. And so, again, we mentioned that, you know, having a strong, trusted partnership is really critical. And as we get this first one up and running, it will make the next disease area even easier. And I'll also mention that as Sutter clinician researchers get their hands on this data and use the platform to really bring it to life and do start that exploration, it really fuels interest in getting the next study up and running. I think we are at the end of the hour. Greg, again, thank you for your time. Thank you for the great presentation and answering all the questions. If those of you in the audience did have further questions or didn't get an answer to your question, Please feel free to direct any additional comments or questions to the contact information that's showing on the screen now. And thanks, so it's Tranah G@sutterhealth.org and info@DNAnexus.com. Thanks everyone for participating in today's webinar. We really appreciate your participation and your attendance. You'll be receiving a follow-up email from XTalks with access to the recorded archive for this event. And a survey window is going to pop up on your screen, and your participation is also appreciated to help XTalks improve webinars. And now please join me in thanking our speaker, Dr. Tranah, and feel free to share this webinar by clicking the link that is appearing in your chat box.
All right, well, thank you, Megan, and thanks to people who stuck around. Have a great day.